So, hi everyone. I think like we're ready to go. Just for the context, is being recorded right now, and I plan to post it on YouTube later, just so that like some other people can access to it and like can look through it. For new trainees, this might be more interesting to you because like if you're new to like plasmids or some other things about like genetic engineering, then I'll try to cover part of those. If there is any question, just feel free to interrupt me. I plan to talk for maybe like 40 minutes and then like, open it up for Q&A, but you can just interrupt me anytime. Just keep in mind that like, if you ask during the presentation, then I may just like, put your question onto the recording as well. So like it might be on my YouTube. If you don't mind, feel free to, just so that like, others can see. But the title that I want to do a mini lecture for today is about plasmid architectures. And I just like put another wordings at like, and utilities in bacterial synthetic biology, which is like what we are doing almost every day here at the lab here. So for the context of people who are watching from the recording right now, my name is Ice. I'm a postdoc at the James Carothers and Zalatan labs here at the Center for Synthetic Biology at the University of Washington. So this is like for our internal kind of training. So if you find it useful, that would be great. All right, let's just get started. So the purpose of this lecture is about plasmid. It's not something that I just make up on my own. It's something that I, just learn across the years while working in these labs and also like working in the synthetic biology. So there will be a lot of resources in like, online out there in the article journal or like even, in the blog web page. So if you are curious about like plasmid or architecture in general, you can take a look at some of these resources out there. I would recommend AdGene, which, a lot of you who may have been working on synthetic biology for a while may know about it. It's a repository that you can request plasmid from, and they will just like give you some description of like what kind of plasmid you're taking from. And there are some reviews about plasmids that would be interesting, and there are some general plasmid kits or toolkits that you can request from the author or the organization like AdGene, so that you can use that for your research. And that's going to be a lot easier than building things from scratch. I mean, we are always like working on the shoulder of the giants. So why not just like take a lot of things that were available already? And for the overview of this talk, I'm going to give a little bit of intro about, central dogma and like DNA replication, because it's going to be very relatable to what we're going to talk about in the plasmid like discussion today. And then we'll go through like what is plasmid exactly. We'll divide them into like the replicable plasmids and also the integrative plasmids, and they are, practically a little bit different. And I'll talk about the parts of the plasmids in the replicable plasmid, because those are something that was like a lot easier to work on compared to integrative one. But we'll get into like the nitty-gritty details of the integrative plasmids at the end, just so that we know what kind of like, why do we need to do the integrative plasmids? Why do we need to make one? And like, what is the purpose of like making this kind of like sophisticated design plasmids? All right, so the background. Hopefully you have learned in biology that like, most of the stuff that we are working on will involve working with DNA. And if you consider like, this kind of like system in biology, it's more, it can be compared to, the system that you work in your computer. So assuming that like you have a computer or a mobile phone or whatever, the DNA that is in the biological system is considered like a storage system where you store the information. Like it's something that determines whether like you're gonna have a black hair, you're gonna have the blonde hair, or it can even distinguish whether like, when during your development, should you have like 2 hands or 4 hands, something like that. And like during those process, the DNA that was recorded as this kind of like hard disk or storage space. Not sure if like if you're too young, you see that those kind of hard disks. But yeah, you can actually like transcribe, which is like, take the DNA, turn into some other molecule, which is RNA, and like use it to do some other processing in the system. And with that RNA, you can translate the language of the RNA and DNA into the protein language. And those protein is actually something that we do be doing the function. And so in this case, it's more about like, you have the computer that store information You take it out from the hard disk, put it into the RAM so that it starts to run. And then at the end, you return into your application. You listen to music, you can watch the video or something like that. The cell is behaving similar to that kind of system. The other thing I want to add is the DNA replication mechanism. So the DNA is double-stranded, and whenever you want to make more DNA, you need to use the DNA itself as a template and just copy it over and over. So you might be a little bit familiar with how the DNA was replicating. So the way DNA replicates is that it's gonna split itself from the double-stranded. Like if you have two threads that was intertwined together, you have to pull it apart first so that you know what is inside the thread of the strand. And then you just take another molecules and try to build up another double strand on top of those split strand. And with that, at the end, you just have another copy of the DNA. It might be hard to look right here, but this is ORC, is written as ORC, which is original replication. In bacteria, there'll be just one spot of the original replication. And at the end, when it's finished the replication, it will just stop at this termination region, and you have two copies. So when you look at it this way, it's going to be very similar to what I'm going to show you next in the plasmid system. Before we go that far, let's talk about what is plasmid. Yeah. When you ask ChatGPT or look into Google, the first thing you find about plasmid is that they're circular. They are probably something that was way smaller than the regular genome or whatever DNA system that is required for the maintenance. Plasmid is something that was replicating independently of the genome. So if you compare the size, it could be you have a basketball that was the size of the genome. You may have a tennis ball that was the size of a plasmid. So the size is usually very different. And most of the time, plasmid is not that necessary for the maintenance of the cells or for the growth of the cells. So just FYI, even though plasmid were well known as a circular DNA, there are some linear plasmids, and they're pretty useful in its own way. If you are interested in that, we can talk about that later. But we will talk about the circular plasmid, in general in this talk. The other things about this plasmid is that if you want the plasmid to be fully functional, you have to make sure that they have all the essential parts of the plasmid. In biotechnology or in synthetic biology, there are only a few parts that would be important for the plasmid to be maintained or to be selected or to make sure that you can use it efficiently. The very first thing is the origin of replication. So this would be pretty similar to the ORC that was shown in the previous picture. You just have to specify where in the linear DNA it should start the reading so that they can make more of these plasmid DNA. The second one is the selectable marker. Most of the time it will be antibiotics, but it can be some other things. We'll talk about that as well. But the selectable marker is something that will be useful when you're trying to see which bacteria or which organism has the engineering performed to them. For example, you wanna see which you have 3 dogs that you have in your in your house, let's say, and you want to mark and make sure that one of your dog has actually ate your pie, So you have to look for the dog that has some leftover pie in the mouth or something like that. So this is very similar. You just have to put some mark into the plasmid just to make sure that you can select for those kind of marking that was done previously. The other things that are not really essential, but could be essential in the specific application you're working on are homologous region. This will be very important when you are doing the integrative plasmid, where you try to put into the genome, you don't have a separate DNA anymore. You just add a little bit of DNA into the big genome, kind of doing some Lego work. Or you can have whatever gene of interest that you want to work with. You can just put it in addition so that when you select for the plasmid that should be maintained, your gene of interest or your foreign DNA or recombinant DNA, those are some common words in the field, will remain and function as it's supposed to as well. All right, so what are the difference of replicable and integrative plasmids? As the name said, the replicable can be replicated, right? And the integrative plasmid, usually we would do integration, where like it just like put itself inside the genome. The image here is, from Wikipedia, and it's something that was like pretty clear when you're trying to show what is the replicable plasmid and the integrative plasmid. If you take a look right here, the plasmid that get into the bacteria, like in this box of things, if they can stay in the cell, like a standalone component, and once the cell divide or just like replicate or just like double into like multiple cells, if they can stay as a circular DNA independent of this genome, that's something we consider them as a replicable plasmid. If it is something that can't really replicate, but can stay in the bacterial cell only if it gets into the genome, so that they can divide and keep these part of the plasmid inside it, that means it's an integrative plasmid. And the easy way to distinguish these kind of plasmids is whether they can replicate or not inside the cell host that we're talking about. So if we go back to the part of the plasmid that we just talked like a few slides before, what this kind of plasmid should be lacking is like the ability to replicate or the origin of replication, which will be dependent a little bit because we talk about the host that we will use it, so that some plasmid can be replicated in only a particular host, so we engineer them to be able to do so. But we just like make some plasmid be unable to replicate in the regular host or something like that. On the right hand side is like an example of the single crossover event when this whole plasmid just get inside the genome. So from what you can see right here is a gene called NICX, where it has like HR1, which is homologous region 1, and HR2, homologous region 2. If they do recombine, either like with this like left-hand side recombination or the right-hand side crossover, it will yield this kind of like DNA construct or this kind of DNA construct, and it get into like the final condition where you can just like take everything back and get to the original construct or have a deletion of this red mark. So that's something that we use the integrative plasmid for. All right. The other things that I want to go into specifics a little bit is that like when you take the plasmid DNA and look at it in your computer, you get the random text of DNA, ATGC, and you'll be annotating them as like, this is part A, part B, or whatever. Most of the time, you see some common parts in those plasmids where they'll be labeled as like ORI, or the original replication. So these are some common parts for the plasmid regulation that you see. Some of the parts below are not that necessary. For example, you need the origin of replication so that the plasmid can replicate. You will need the host to have the particular protein. For example, DNA A sounds like a pretty good abbreviation, because like it's DNA, right? It's gonna work with some DNA for sure. It's actually a protein for DNA replication inside the bacteria, and you need some replication proteins. So that rep protein, it might be like rep A, rep B, rep C, or whatever number or alphabet that were given to it. It depends on like the class or the group of those plasmids. And most of the time, it will be involving with the initiation process. So you usually need those rep protein in order to make sure that the plasmid can replicate properly. But there will be some other things like the COP something, BOP something, and PAR something. Those would be relevant for the copy number control, the mobilizability, which is like governing whether the plasmid can like travel among the population or something like that, or the partition control, where it can help when the plasmid is actually segregating from each other. Like if you have like 3 plasmid in the bacteria, you want to make sure that the bacteria, when they divide into 2 different cells, they take one of the plasmid, rather than like having one daughter having all 3 plasmid and the other one has no plasmids. That was like the other system in the plasmid that help with the partitioning. But most of the time, those 3 additional things can be found in the plasmid, but in the biotechnology application, sometimes we just omit them so that we don't have to include too many DNAs into a plasmid. So let's in deeper into how the plasmid replication actually occur. There are 3 general types of replication that I found in the literature that I'm looking at. So the first one is called theta replication. And you'll be a little surprised why do they call it theta? Because if you take a look at the picture on the right-hand side there, it actually looks like this big theta in the Greek character. So the rationale behind this theta replication system is because of the shape that was found. When you look into the microscope, you see this theta character inside it. And the way it works is that like it may require some rep proteins if it doesn't have the rep protein inside the cell already. And it may have some iterons. Iterons mean like the repeating part where the DNA binding protein will find it. So like if you look into the Plasmid Replication Journal, you see a lot of terms that mention rep protein and the iterons that will involve with like the original replication in the system. So as you can see here, the figure is very close to like what you've seen in the like DNA replication in the genome, right? It's basically like almost the same, where it's just like split apart first, making some theta looking, like making some loop right there, and just keep extending over and over. So this is the experiment called Kane's experiment, which is pretty famous. It's an experiment where they label these DNA with some radioisotope, and they try to take an image over the time. So it might be a little bit difficult to look. I'll share the slide later, but the image that we have seen here is similar to like the drawing above. So it actually formed some loop right here. The loop will get bigger and bigger, and doesn't separate from each other at all until the end, where there are like two loops connected together, and they're just like split apart at the end. So this is the theta replication. Again, this picture right here, this molecule right here, looks like the letter theta in Greek. That's why. Right. The other one is called strand displacement, and the mechanism will be pretty sophisticated. It actually requires like these two SSIA, SSIB RNA to work independently of the host, and they usually require like more replication protein to help with them. So apparently, like one example of this strand displacement is the PRSF1010, and it's a pretty broad host range plasmid. So most of the time, if we talk about the host range, which I'll touch into in depth later as well, if they have like a lot of replication machinery like carrying over in the plasmid, then they usually rely on the host much less, because like they were using the proteins or the other things that were inside the plasmid to help with the replication or the maintenance. So that's how the plasmid can stay or can mobilize much better regardless of the host. So this mechanism is one of the key, like attributes or parameters that control the transformation efficiency and like other factors. So when we talk about something that was like high copy or low copy, which I'm going to touch a little bit later as well, these are the mechanism that was involving in whether like you're going to have like an efficient or inefficient transformation or maintenance of a plasmid. The last one is called rolling circle mechanism, which is mostly found in gram-positive bacteria. Again, if we talk about E. coli or like some bacteria that we're working, in general in the acidic biology labs, we usually work a lot more with the gram-negative bacteria, like especially E. coli, and a lot of gram-negative plasmid doesn't work in the gram-positive bacteria. So there'll be some ways that we have to work on that. And most of the gram-positive bacteria will use this kind of mechanism to replicate the plasmid, which is called rolling circle. And the difference of this rolling circle is that like it just start reading at one point and just go over the ring, and then stop only when it comes to the final one already. And then this thing will get replicated again. So the main difference is that you're not generating two separate plasmids like right away. You actually could generate like one half of the plasmid, and then the other half of the plasmid will be generated afterwards using the first cyclic DNA that was generated here. And it's called rolling circle because it just keep rolling in the circle. One similar things that what was found in this kind of system is that like it was known that like it's a lot easier to have multimer or like multiple, like a bigger size of plasmid when you read and you don't actually stop, just keep rolling and rolling and rolling, and you ended up with twice the amount of size you ended up like putting too many Lego blocks into the PC you want to build, and you just have twice the size, something like that. All right, those are mechanisms. And with these different mechanisms and like plasmid out there, you can actually make the toolbox. So I'm gonna talk about two main toolbox that we've been using in the lab and maybe like in the world as well, because like when you have like some systematic system that you can segregate or like organize these kind of tools, it's gonna be a lot easier. One thing that was like, came up during the emergence of like the field of acidic biology is called the BioBrick or the BioBrick systems, where they have like the BioBrick compatible parts right here. Here is going to be like, different repressor for inducible system, which have like 4 systems that you can choose from here. There are actually 8 types at the end, because like there are some different promoters that control how much protein you want to express from these plasmids. They have like different cargo, and in most of these too, they just use protein for ease of like visualization. So the bacteria turn green or turn red depending on like what is carrying. They have the replication origin that you can change them according to like the copy number or the host range you want to work. So the start of this like system has only one broad host range that can work in multiple organisms in here, and the other ones are just like E. coli plasmid, and there are 3 of them. And the antibiotic resistance that was available in this plasmid toolkit are 3. And you see that like the numbers that were given here is just like a few or a couple. And the day and the year that this system came out was like 13 years ago. And that means like that's the best we can do at the time, maybe. So we have been using this plasmid when we start the lab. I believe we have some of these PBB vectors that we keep using, that we keep dealing with. But after the years, other system that I've found to be like much more systematic, because it's gearing toward the multi-organism system much more. So this system is called SIVA, or Standard European Vector Architecture, where they have like more defined enzyme cut sites around here. There are some enzyme cut sites in the BioBrick system as well, but this one is a little bit more systematized, where they take it in consideration where these replication system will have the same direction of the replication protein, and they will have like the orientation of this readout in a more systematic way. They also take it into consideration if you want to make a shuttle vector, which I'm going to get in a little bit later, on like having like more than one origin of replication in the system. So what you can see here is that like they actually have more antibiotic resistant genes available. I think they have up to 5 when they first like make this system, because when you were trying to work with new organism, sometimes they are already resistant to the antibiotic or the selection marker that we're using already. So we have to like find more and like make more antibiotic resistant available into the system for ease of like manipulation. And for this one, they have 5 replication system available. Again, reminding you back that like some of the system has only the origin part and doesn't have the protein part for the replication. And that means that it's going to be a lot smaller, but it may require something from the host to make sure that it can replicate. For PBBL1 and PRK2, you see that you have like both ORIV, which is like the original replication, and also the rep protein. In this case, it's like rep of the PBL1. In this case, is TRFA for the PRK2. So it depends on like which type of plasmid that you're working on. For this one is a shuttle vector, and you can see that there are like something similar to the PBL1, where there is like ORIV and rep, but it has this CoE1 origin as well. The reason is because like this replication system is only applicable in Pseudomonas, not E. coli. Pseudomonas is an organism that is a lot harder to extract DNA from. That's why you want to make a lot of DNA in E. coli first, and then put into Pseudomonas later. And that's the meaning of the shuttle vector, where you can have the manipulation in E. coli because it can replicate in E. coli. And then it can also function in the other organism that you're interested in. The last one is PRSF 1010. And you can see that, like, as I mentioned, that like the replication is a lot more complicated, and it actually has 3 replication proteins. so that they can start their replication system properly. The size right here is relative to the real size or the real length of the actual origin. So you can see that like the R6K is a lot smaller than the RSF1010, and that will control, some downstream application as well. Like the bigger the backbone is, maybe it's going to be harder to maintain or to replicate. One important thing for this SIVA system is that like they take conjugative transfer into consideration as well, because when you're trying to transform some DNA into the new organism, it could be hard to generate the new system that can transfer the DNA into the organism of interest. And it was known in the past that like the conjugative transfer is the method that allowed the maximum contact time or the maximum, efficiency in terms of like if you try the transcription for the first time, the conjugative transfer or bacterial mating is actually the process that is that will give you the highest likelihood of being able to transform. So this is the key difference of the SIVA system and the BioBrick system, in my opinion, where you can have this like origin of transfer for the conjugation. And you can also like change this origin of transfer into some other thing if you really want to optimize that process. This is a little bit about like the transformation efficiency and conjugation. So most of the time, if you have higher copy of the plasmid, you have higher transformation efficiency or a higher likelihood of like being able to put the plasmid into the desired organism. So it's pretty straightforward, I believe, because like thinking about the rate of the plasmid that will be generated. If you have some plasmid that has like only one copy, it means that like it's going to be pretty slow in terms of making a lot of proteins or like making a lot of plasmid, right? Compared to the plasmid that will have like 1,000 copy numbers. It means that like you have to ramp up the production of the plasmid a lot inside the cells. So when they get into the cells, they will just like divide themselves or like replicate themselves so fast. And that means like you're likely going to have like a higher transformation efficiency compared to something that has like only 1 or 2 copies, and you have to take time to make sure that the selection or the protein that they're needed for the selection is well expressed. That means like there will be some that actually have the plasmid, but doesn't survive the selection that we are applying to. There are also some other mobilizability or mobilizable genes that are relevant to this system. The MOB-AB is one example from the PBBL1 original plasmids. Sometimes it was omitted from the system, like the SIVA system doesn't include the MOB-AB genes or the mobilizable genes A and B anymore. Likely because they just provide ORT, where you can use that with the other system already. So, sometimes you don't really need all the parts of the plasmid, as I said. And this is like, an example of like how the transfer of DNA occur. They will make some pili, like some tube that connect two bacteria together and then transfer DNA along those tubes. So this is a pretty clever way of the bacterial conjugation or bacterial mating where they share their genetic materials together. All right. Apart from having different plasmids, we also have to think about, the plasmid compatibility. So if we have, one plasmid and we try to put into bacteria that doesn't have any plasmid before, then we can probably, select for the group that has plasmid or doesn't have plasmid, right? That's pretty easy. But when we talk about like the bacteria that you want to put multiple plasmids or multiple pieces of DNA into them, you want to make sure that those two plasmids are not interacting with each other too much to the point that they don't allow, their peers to stay. Assuming you have 2 tigers in the cave, maybe they'll just fight until one is dead. There's like a result competition all the time. But if they're, if they can coexist, then that means you can keep 2 plasmids, for example. So in this case, one good example is in E. coli. If you have like the PMB1 derivatives, which cover like the PUC, PAP plasmid, or CoE1, or PBR322, if you're working with these plasmid derivatives and you try to put the same plasmid system into, the same cells, then they will likely share some replication or mechanism with themselves. And that would be harder to make sure that those plasmid will remain as you program, something like that. But you mix like this plasmid group A with group B, or we call this plasmid P15A, or with the group C, which we call them PSC101, they can be mixed and they can just stay independent with each other because they have different niche. They're using different kind of tools and machineries to make sure that they can replicate in the bacterial cells. The other thing that would be good to keep in mind is the host range of the plasmids. Like you're trying to put the plasmid into, particular organism, maybe they cannot really interact with the host replication machinery, and that might mean that they cannot replicate or stay in that organism efficiently. So again, back to like the when we talk about the parts, the plasmid that have more replication machinery installed into the plasmid will likely have more likelihood of being independent or being able to like replicate or stay inside the cell much better. However, there are some examples out there when scientists are trying to make sure that we can use the, I would say like turn the bug into the feature or something like that. You can actually take the plasmid that have the same same replication origin and just mix them together and see if you can segregate or like control them to make sure that the population A and population B will be maintained and like can change across time. So this is a example of the paper that was just published this year about trying to put the same CoE1 based plasmid together and see if the resources were shifted towards the other condition, then the amount of plasmid will sort of change as well. The ratio of plasmid A and plasmid B will sort of change depending on the environment. So again, this is sort of like, the kind of like burden and competition and other things that was used to control the population of this system. The other parts that we talk about are selectable markers, and I hope that so far we may talk about antibiotic a little bit, but selectable markers doesn't really mean only antibiotics. So there might be some other selection that you can put inside the bacteria and make them behave better or like be able to grow faster or something like that, so that you can isolate the one that has your gene of interest over the one that doesn't have your plasmid or gene of interest. So in this case, there are two main things, which is like orthotrophic. Orthotrophic means like something that you need it in order to grow. For example, the bacteria are actually capable of making all 20 amino acids that we have. Like human cannot. Human need to like eat like other organisms to get amino acids from them. But these bacteria or yeast or other organisms, sometimes they have all capabilities to make these amino acids. But you delete some of those genes, you can actually make those organisms be orthotrophic, so that like they have to grow only if you supply these nutrients. And that was like one of the strategy where you can reintroduce or select these organisms using this kind of extra orthotrophy. The other strategy is maybe using the toxicity-based selection. So think about like you're feeding the bacteria with poison simultaneously, and it will die if you don't give them the antidote. It's somehow work like that. If you transform the plasmid into the bacteria that were suffering from a certain disease, and you give them the antidote on the plasmid, then the bacteria that grow after that can grow at the end is only the one that we're recovering from the antidote. Or vice versa, you can also transform the plasmid that has these like toxic compounds or like ability to make toxic compounds, and then like feed the compound that was not toxic yet but can turn into a toxic compound only if the genes that can convert it into the toxic compounds exist. In that case, you can do counter selection, where you can select for something that doesn't have plasmid instead. So these are like some common system that has been used in the field so far. These are the list of like general antibiotics that were available, as I've seen in the SIVA system at least. So the list right here on the right are like 10 of them, and the three counter selection method or like toxic system listed there should be pretty common in the literature to some extent. So you can look further and search for this compound. You should be able to see if they're applicable to the organism you're working on or not. Again, it doesn't mean that these systems will work as magic for every organism. Sometimes the particular bacteria has resistance to a particular antibiotic because they're just special. So having more of these tools available will always be beneficial if you're trying to do a particular application that you don't really care what kind of selection you will be using. Right. This is like a good example of like the auxotrophic system. On the top is an auxotrope, and on the bottom is a prototrope where they can just like make the arginine amino acid by itself. So you can see that like if you have the minimal media, the arginine auxotrope cannot grow. Only if you supply arginine, then they can grow. But if you supply just lysine, it doesn't matter. It's not the right antibiotic that they need to grow, something like that. So if you supply the arginine, instead of supplying arginine, you supply arginine producing genes on the plasmid instead. Then you can actually like let them grow. On the right hand side is a relatively recent work from this group, from Tassoc Moon group, where they can actually like make this antibiotic free antibiotic resistant gene free plasmid by engineering E. coli so that they lack some essential genes and introduce the essential genes into the plasmid instead. So this is like similar to the auxotrophic system, but it hasn't been made a lot in the past for bacteria. But this would be beneficial in a way that like if you are actually making a lot of antibiotic resistant DNA in the group, and you want to make sure that you can dispose them 100%. That's pretty difficult because DNA is a pretty stable molecule, and you don't really want to throw DNA and like disperse them into the environment. So not having them at all will be pretty safe if you think about the containment side. And it would be helpful in terms of like you don't really want to express like some additional extra genes that you don't really want to as well. All right. Now, diving deeper into the shuttle vector a little bit. So I try to use this figure. I'm not sure if it's going to show up on the recording, but think about we have like, a plane or a bus, and we want to like make sure that your plasmid can function in both like the organism that behave like a bus or the organism that behave like a plane. Then you want to make sure that they have their parts that are applicable to replicate or function in both organisms. So in this case, these are like some example of like the original replication for non-E. coli systems. And if you actually combine them with the E. coli replication mechanism like the CoE1 system, you can actually make the shuttle vectors. If you use something from yeast, you can also make the shuttle vector for yeast and also for bacteria as well. And the same goes for like the other system. These are something pretty common in the field. The only downside is that like you may have some additional parts of DNA that was, important for the maintenance in E. coli, which is an easy tool to work with, and it will be useless when you put it into the new organism. But sometimes those like 500 base pair is not that bad to keep it around, right? So most of the time, the shuttle vector will have like not just the replication mechanism for the E. coli itself, but it may also have the selection marker for the E. coli, because if you change the organism into the gram-positive or the other kingdom of organism, they may require or like prefer different kind of replication, or sorry, they may prefer different kind of selection or selectable markers as well. So sometimes you just have like two markers and two origins, and they're just shuttle that can work in either way. If you have all the parts all together, you can actually turn them into a molecular cloning toolkit. On the left-hand side is a toolkit that people in the yeast have been using all the time. And on the right-hand side, it's a tool that has been recently made and was just published as a archive. So it means that like making these tools are always useful to a certain extent. If you don't have the tool that will fit perfectly to your work, then you may need to make the new one to make sure that like, your team or like your company or whatsoever can work faster in this environment. This new B subtility system is important in a way that they can make the multi-operon system very rapidly. And multi-operon means like putting the same gene onto the same transcription factor, so you don't really have to separate them that much. And it was designed in that way. So you can just like make these within like a few days rather than like trying to make something through multiple weeks and have like a risk of like failing it as well. So these are like, the outcome of the, having a lot of toolkits or like parts available in the stock, so that you can like plug and play, pick some different Lego parts and like combine them together. All right, so finally, it's almost the end of the talk, but like I'm gonna touch about the integrative plasmid very briefly, because like some of you may be interested in using the plasmid tools for the genome manipulation systems. So one common thing that it might be a little bit difficult to follow in the genome integration or like the integrative plasmids side is that like there are two ways of doing recombination, like or the classical recombination, which is like the single crossover or the double crossover. So this figure is a pretty clear narrative for you to follow what is the single or double crossover. So take a look here. You can see that like considering this like shading as a crossover or whatever like place that will replace things inside the plasmid and in the genome, the target genome. You see that like in this case, they have only one region that was highlighted. On the right hand side, you have two regions, and when the replication, when the integration process get further, then you take either the whole thing in or only part of that in. So if you do double crossover, most of the time you only take part of the plasmid and integrate it into the desired target genome or whatever. But if you do a single crossover, it will get in as a whole piece of circular DNA. So there'll be, these will sort of like control the design or whatever downstream manipulation that we have to do. So let's go to some example. If we use some like fancy techniques to deliver DNA or part of DNA into the genome, they will use like three examples of like machineries or system. First is a transposase. Transposase came from the molecule group called transposon, where it's actually like move around inside the genome or in the bacterial community or something like that. The thing about transposon is that like it's composed of like different proteins or different machineries. Think about they're just like some thief lurking around in the community, and they're just trying to like sneak into your genome whenever they have the chance. So there are different types of transposon out there. There are some transposon that like prefer one particular location of your like organism of interest. There are some transposon that doesn't care anything about anything at all and just want to hop into the genome randomly. So the way it works is that like there'll be this part called like the transposon iteron region, where the protein itself will just come and take the part between these two recognition sites or level, and we just like loop this part up and put it into the final target DNA that we wanted to. So in fact, this is how the transposition occur. They just take part of DNA and put into the target DNA. So in this case, it means that like you probably don't really need the whole plasmid to get into the genome, right? If you have just like the part of the DNA that has these like recognition site on the left or on the right, then that should be enough. But you need like the whole transposon machinery, which is like the carrier of this DNA to put it in. The other one is the integrase. So most of the time, the integrase will depend on the protein that will work on the integration. And the other part, which is like the recognition site on the donor and the recipient part. So an easy example right here is from the phage system, where there was like ATTP and ATTB, which is the recognition site of the host target, and the phage DNA, which is like circular at this time. If you do the integration, what is going to occur is that it will get into the genome as a whole. So like this whole thing, ATTP, we get split by the system, and inside the cell in between these ATTP, and this side will just be different. It will just be like, you know, A1A prime, B1B prime, change to AB and B, A prime, B prime, or something like that. So the thing to consider here is that like even when you use integrase, you have to throw the DNA in as a circular DNA. If you throw in the linear DNA, it cannot do this process because the resulting DNA would just be something that was like broken. And the bacteria that have broken chromosome cannot really grow properly. And like with these tools, if you use like phage integrase or serine integrases, you can actually make a toolbox. And this is called serine integrases assisted engineering, or SAGE. It's pretty interesting as well. It can be used in multi-organism so far. And the final one is the part that was based on recombinase. So for the recombinase, it could be the tools that were specific to like some homologous region. You may require like a thin link of DNA so that they can recombine and like put themselves into the genome, something like that. But there are some other tools that are specific to the size that you're looking for. For example, the Cree recombinase. So these are some example of like some Cree lock system that they can do like, not just integration, but they can do inversion, like flipping the gene. They can do deletion, they can do translocation by like moving parts from one to the other. These are going to be like more useful when you work with higher organism, when you have like multiple chromosomes, because if you have more chromosomes, you can move from one chromosome to the other chromosome way easier. But if you talk about, if you focus on the bacterial part that has like only one type of chromosome, you can actually like make the target site having the specific recognition site, and you can just put your piece of DNA that have those sites into it. And after they found those sites, they'll just like replace or like integrate the piece of DNA into it. So the good thing about this system is that, again, you can just put in as a linear DNA as well, or you can put it in as a plasmid, because the recombinase will just come and pick only the part that they like and put just that part into the genome. If I were to draw it, it's going to be like this. You need like a part of a plasmid or linear DNA for the transposase to work. Of course, you need some like transposome complex. If you want to work with the integrase, or at least the integrase that was like mentioned in this talk, you have to put it as a circular plasmid most of the time. But if you use recombinase, it's going to be similar to the transposase, but most of the time it have to be more specified, not random. Like on the top part, the transposon system have some like random integration as well. So that's like one of the cool things of the transposon. And I think that's it. This is what I want you to take away. Make sure that like if you want to use this DNA or the toolkit for anything, you have to choose wisely, depending on the system you want to work with or the organism you want to work with. And that means like you have to choose the plasmid backbone properly. Sometimes you need the shuttle vectors. You have to choose the selection markers and the method of transformation efficiently. Otherwise, you may not get, the engineering that you wanted to. You also want to make sure that your microbes, if you were to put multiple plasmid into it, they can carry those two plasmid efficiently. Because if you just try to like randomly put two plasmid of the same origin in, they will compete for the resource. And we even have like some ideas or some work out there that we're using that idea to show the resource competition for real, right? And at the end, some of the high throughput methods for the genome engineering may require like some sophisticated or pre-designed plasmid so that it works as you want them to function. Again, there will be different scenario when the things get in as a linear plasmid, as a linear DNA, or as a circular plasmid, or whatsoever. So most of the time, if you want the system to work specifically or more efficiently as you want them to, you may have to turn them into a very well-defined toolkit or system that you can work, so on and so on. And maybe the final things is that like it's worth it to revisit what is available in the team or what is available in the field first before like moving further and try to make a lot of new things on your own, because like maybe like it was already available and you don't have to reinvent the wheel and just like go ahead and make the car instead of the wheels. So that's going to be a lot more faster than like trying to do everything on your own. I think that's it. Hopefully that's helpful. Any question? Clappings out there. All right. Any question? Hopefully it is not too long. I just like put random stuff all together that was relevant to the plasmid, I believe. There was one question from Diego. Diego said, but sometimes we just like making wheels, and that's true. Because if your wheel is not good enough, just make a better one. All right. Any question?
Like now, for how we plasmids, is there like a sign where like you should not go over? Basically like or like certain couples have too many cellular resources for replication. Like for fully one, like is that kind of like, do we start thinking about like capping our plasmid?
Yeah, good question. So the question is about is there a maximum or preferred size of the plasmid that we should set a threshold on? Especially if you're making a lot of plasmid in a high copy number, make a lot of plasmid inside the cells. I think the answer from the field is that like the copy number of the plasmid is not something definite. It depends on the size of the plasmid and also what is on the plasmid as well. There are a lot of studies that like tell us that well, if your plasmid is taking more resources to make, for example, the plasmid is bigger and it takes longer time to make, 20 kb plasmid rather than a 3 kb plasmid, then it's likely that the copy number or the amount of plasmid itself get decreased. So even though, yeah, sometimes it was like kind of self-regulating in a way, because like if we try to put them into, if we try to put them into the equation, right, it would be like the production rate or the replication rate of the plasmid. And if we dig deeper into the rate, it might be the rate of like replication per base. So if the rate of production of the per base is remaining the same, but the production rate of the whole plasmid is taking longer, then you will likely get less number of plasmids. So there are some models that are like hopping around, but there are more things involved with this system, right? Because it depends on the starting material that was available to make these plasmids. So if they take up a lot more resources, even though they are the same size, they may just have lower copy number as well.
I have a question about doing a dual plasmid system. If you have like a high copy number of plasmid and a low copy number of plasmid, assuming that the high copy number of plasmid will have more antibiotic resistance within that system, does it make sense to dose that organism with a higher antibiotic than the low copy?
Yeah, that's a good question. So the question is about if you have two different plasmid copy number, like different amount of those things, do you have to change the amount of antibiotics or selection marker or molecules that you're using or not? A good answer would be sometimes you don't really have to change, because when we use this selection is about like whether you kill the bacteria or the organism that doesn't have the plasmid or the resistant marker at all, and the one that has one. So the effect of having higher copy number, of course, will make sure that you have like more resistance towards that thing, but maybe you don't really need to add too much of those selective molecule, because only, let's say, if you're trying to select them by putting chloramphenicol into it, if 5 microgram per mil, like 5 unit, is enough for selection already, then maybe you don't really need to add more, because adding more means like you're using more resources and the likelihood of like things like escaping might be higher as well. And most of the time, the way that the bacterial fighting with these molecules are dose dependent, and adding more doesn't always help. There's always a chance that like you can decrease them. Let's say if you are using like 2 antibiotics for selection and you've been using like the regular concentration for the selection so far, and you can't really get these things to transform effectively. And it might be because one of those plasmids were expressing with the less number, a copy number or something like that. So you ended up with less enzymes or protein to fight with this selection marker. Maybe decreasing the antibiotics or the selection molecules will help with the selection as well. But this depends a lot on like the step that you're in. Let's say you're trying to select for the one that has these traits or has a plasmid, then you might be able to like decrease the concentration to a certain extent. But whenever you come back and try to make sure that you maintain the selection, I think people in the field usually prefer to like add sufficient amount of the selection pressure. Otherwise, you may find something that spontaneously get resistant or spontaneously ignore the presence of this selection because the selection pressure is not too strong. Yep. Any other question? think the timing is okay. All right, it's recorded, and I'll share the slides. If there is any other question, then it can be, point out at the recording instead. All right, thank you, everyone. I'll stop the recording now.